It's my great pleasure to introduce our work from mechanism to application, decrypting light-regulated denitrifying microbiome through geometric deep learning. Denitrifying microbiome play a pivotal role in both nature and society. Optogenetics is a promising regulatory strategy on them. While decrypting biological big data, especially meta-omics, is facing great challenges, deep learning has exhibited superior performance, especially the graph neural network and the topological relationships. There are three aspects of highlights. First, we use biological contextualized GNN. Second, we validate the results in wet lab. And third, we develop different toolkits that everyone can use. As for the results, we found the light-regulated microbiome metabolisms. Blue light exhibit inhibitory effects on microbiome, while yellow light boosted both nitrate and nitrate removals. In comparison, blue light reduced biomass accumulation, but no significant reduction in carbon source intake, which implied the metabolism flux diversion. To decipher the biological big data, the knowledge-based database classification failed to obtain coexpression panels. So we use GNNs to decrypt the meta-omics, especially for the microbial interaction and the extra and intracellular substance exchanges. Here are some details. We extracted the differentially expressed genes and integrated the expression information and the subcellular information and employed Deep Graph Informatics (DGI) to obtain the gene panel. We evaluated the clustering results obtained from the geometric deep learning. The mathematical validation, it turned out that DGI exhibits superior capacities in integrating heterogeneous information. And on the perspective of biological validations, it also turned out that DGI could better identify genes for certain functions. Based on the gene panel we obtained, we can use them to predict phenotype. That's the first toolkit we developed, the coregulation panels for new discoveries. For example, the Hopper gene panel of yellow lights has a lot of pathways that enrich related to peptide and protein synthesis, and the secretion system were also upregulated. So in the wet lab, we validated the protein production on the yellow lights was extremely enhanced. So that for the blue lights, that is the biocatalytic effect of EPAs, is also validated in the wet lab. The second toolkit is topological network with landmark genes for regulation strategies. For example, under the blue lights, we can see that superoxide dismutase is the landmark gene of nitrate reductase. So in the wet lab, we found the co-regulation between superoxide and nitrate reductase. Here are the mechanisms. Overall, secretion system is the core of cross-species interactions, and the light-dependent denitrification is due to the decentralized metabolism fluxes. And on the blue light, there are some active substances like cofactors and vitamins that imply the potential of utilizing nitrate as substrate for high-value resources. What's interesting is the superior activation effect of yellow light. This might contribute to the cross-species interaction, especially due to the PLS-related proteins. This results imply its huge potential use yellow light for taproot and polypeptides production. So here's the summary of the DMLA cycles. At the discovery stage, we utilize the optogenetic-based genetic perturbation to obtain the dynamic network. And at the second modeling stage, we integrate the expression and the subcellular informations to help the model learning the domain knowledge. At the third stage, the learning stage, we have developed different apps used to unveil unknown phenotypes, elucidate biological mechanisms, and develop regulatory strategies. So this comes to the fourth stage, advancing our biotechnologies. We can use them to domesticate and regulate real bacteria for diverse and versatile utilizations.